Hello, my name is Richard Moore, and I'm a research scientist at NASA's Langley Research Center in Hampton, Virginia. Today, I'll be talking about some of the measurements that we made of particles or aerosols in the atmosphere during the 2019 FIREX-AQ field campaign. And so FIREX-AQ was a mission targeting the impact of smoke and fire-produced emissions on air quality. And our strategy was to sample these emissions using NASA's flying laboratory, the DC-8, which is shown parked here at the airport in Boise, Idaho, under what is a beautiful sunset. And so we saw a lot of beautiful sunsets during this project because the air was so full of the smoke aerosols that were scattering the light. And so before I begin, I just want to acknowledge my co-authors and collaborators at the work, many of whom are at NASA Langley, but we also had a diverse group of collaborators from universities, including Colorado and the University of California, Irvine, as well as other government scientists at NOAA. And so FIREX-AQ was a joint NOAA-NASA field campaign targeting two very different types of fires, and we based in two locations in order to reach these fires. The first were the western wildfires, given by these red flight tracks. And here we based out of Boise, Idaho, as a central location in order to get to these western wildfires. And so these are the large forest fires that are making the news more recently in the past few years that tend to be very destructive both to the forest land, but also to homes and neighborhoods and that are mixed in with some of those forested areas. And so these tend to be very large. They're easy to see from space, but we don't have a great understanding of what the emissions look like for these fires and how we can relate those back to the fire behavior. In addition, we based out of Salina, Kansas as a central location in America's heartland, targeting a very different type of fire, agricultural burns that farmers prescribe after the growing season when their fields are full of crop residue and they want to clear out the field, return the nutrients to the soil so that they can start anew. And so these are much smaller. They tend to be very well controlled, and they're typically about the size of several acres, so the size of a farmer's field. And that makes it a little bit more challenging for us to see these fires from space and certainly their emissions, but they're no less important because when you start to add up the totality of these prescribed burns, you start to see a real impact on the regional air quality. And so we wanted to understand how the cumulative impact of these small fires influences air quality. And so we talked about two different types of fires. We had two very different airborne sampling strategies. And so this is a typical sampling strategy for the western wildfires, which were very large plumes. And we were able to really spend a lot of time sampling the plume from a number of different perspectives. And so the graph on the left shows the flight track in gray. And for areas where we encountered the smoke plume, I've colored that flight track by the carbon monoxide mixing ratio. And the fire, as detected by the satellite, is given by the blue X. And so initially what we would start off, we would be able to very clearly see the plume. We would approach from high altitude, and we would make a measurement using the LiDAR remote sensor of the plume height and intensity along the track. And then we'd loop around, we'd do an above fire upwind leg, and then we'd do a series of these transects perpendicular to where the smoke was in order to get a cross section of the plume horizontally as it ages, as it moves along. And you can see the carbon monoxide mixing ratio begins to drop off due to changes in fire activity over this time period, but also because of increasing dilution as the smoke mixes with the surrounding air. And you can see this on the right graph. Now this is altitude as measured by the LiDAR versus time as the aircraft is flying along that longitudinal LiDAR leg. And the coloring reflects the backscatter, the amount of photons or light that's backscattered from the incident laser beam back to the detector. And so we can see very clearly as we're looking down from above this plume that we initially have a severe or significant injection of the plume up to about 6 kilometers. And then over time that plume mixes down into the atmospheric boundary layer. And so we're able to really get a really comprehensive picture of the fires using this sampling strategy. We can contrast that with the agricultural fires, which are much shorter and much more difficult to measure in a comprehensive way, especially given the fact that we have altitude restrictions in these populated areas where the aircraft can only fly so low. And so in a lot of cases, we had trouble actually sampling the plume because it didn't rise high enough for us to sample it with the aircraft. But our goal here was really numbers. And so this yellow trace represents the particle number concentration that we measured instantaneously on the aircraft as we moved around and tried to target different fires in the Mississippi River Valley. And so there were a lot of these fires. We needed to come up with a system to keep track of them. And so on this particular day, the scientists were calling out alphabetically names of picnic items. And so you can see perhaps the largest plume here was associated with the chips fire with its nearby dip relationship. And then we also saw some more significant plumes for Frisbee, hamburger, and Jello. But this was a very different sampling strategy as shown by the number concentration time series. Here we were in the plume literally for seconds. And so this is where having fast time response CPC measurements was really helpful for getting at the structure of the plume and making sure that we captured the full extent of the emissions. And so in order to support FIREX-AQ, the Langley Aerosol group provided a number of different instruments. We were responsible for overseeing the community aerosol isokinetic inlet that's shown in the image on the top right there, where we would draw air into the aircraft as gently as possible. We would make sure that the flow that was entering through that tube matched the velocity that the aircraft was flying at. And so then we would distribute that flow to other teams in addition to ourselves. We made measurements with condensation particle counters of the aerosol number concentration, both with and without a thermal denuder that was used to strip off some of the more volatile parts of the aerosol. Similarly, we made measurements of the aerosol size distribution using a scanning mobility particle sizer and an optical sizer, the laser aerosol spectrometer. We made detailed measurements at multiple wavelengths of the aerosol scattering coefficient using nephelometers and the aerosol absorption coefficient using a filter-based PSAP. And then in addition to the measurements that we made inside the fuselage, we also had wing-mounted cloud probes that were giving us measurements of droplets or large aerosols greater than about 2 microns. And so on the bottom, you can see the layout of the rack. We were position number 11 on the starboard side of the aircraft, where the forward of the aircraft is to the left. So this is a view of what our rack typically looks like. You can see Dr. Liz Wiggins and Mr. Eddie Winstead operating the instruments. And you can see that we have to be very careful about distributing the flow across all of these different instruments using transport tubes to ensure that the particles that are coming in from the ambient atmosphere at low pressure aren't impacted by leaks or other contamination from the cabin, which is pressurized at a higher pressure for our comfort. And so this is an example of the flow diagram for the Langley Aerosol group. We're operating that isokinetic inlet. We have a common dilution system, and we're distributing sample flow to over 20 different aerosol instruments, about 15 which our group is operating directly, and then also to our collaborators. And I won't get into too much detail other than to highlight a few interesting components that we did specifically for FIREX-AQ. And so for the first of these, we developed a community bridge diluter system. And so many of the instruments under very high particle loading actually saturate because, for example, the particles are detected by looking at laser pulses of light in a particle counter. And so at high concentrations, you often have more than one particle entering the laser beam at the same time. And so that particle or that pair of particles would be counted as one large particle. And so we would be both incorrectly sizing and incorrectly counting the particles. And so we had to develop a system that Dr. Wiggins developed using where we route some of the flow through a filter and some of the flow through a capillary. And we then remix the flow back together before sending it to the instruments to make sure our instruments stay on scale. In addition, we took detailed, in addition to the online measurements, we also collected aerosols on pre-baked quartz filters. So we would buy these quartz filters in and put them in stainless steel holders. The quartz filters were initially baked to 900 degrees in order to remove any sort of volatile organic species. And then we could send these filters to our collaborators at UC Irvine in Jim Randerson's lab to look at the carbon and aerosol nitrogen on the filters and use radiocarbon isotopes in order to tease out differences in fuel age or the fire consumption. In addition to our standard suite of particle counters, the CPCs and the optical particle sizers, we also used a thermal denuder in order to strip off the volatile components of these particles to look at changes in the number and size associated with the non-volatile fraction of the aerosol as well as the volatile fraction. And for FIREX-AQ, something we don't typically do is we also measured carbon dioxide on the aerosol inlet. And so this was in collaboration with the NASA Langley DACOM group, who makes the primary carbon dioxide measurement on the aircraft for these missions. However, the DACOM inlet is on the other side of the plane. It's on the port side of the aircraft, a few window blanks forward of where we sit. And so we had a question about whether or not there was enough heterogeneity in the smoke plume that we would actually measure a different carbon dioxide concentration from that group. And so generally what we found was, despite the difference in spatial location, the aircraft is moving so quickly through the atmosphere that the two carbon dioxide observations perfectly agreed despite these differences in inlet locations. And then finally, and this is something standard that we do for all missions where the large group is operating the aerosol inlet, we employed a inlet filter and polystyrene latex sphere nebulizer assembly. And so what this allowed us to do was, while we were doing flights, we could do periodic instrument zeros or introduce these NIST traceable PSL size standards into the inlet to all the instruments. And this really ensured that every flight we were able to assess reliability and consistency of the instruments. And so, just to summarize, FIREX-AQ provided a really comprehensive data set of smoke from both western wildfires as well as the much smaller agricultural burns in the southeast US. And we deployed an extensive suite of aerosol, concentration, sizing, and optical instruments that's going to result in a number of really exciting future publications on the smoke properties coming from these fires. And I'll just highlight a preprint from Liz Wiggins located at this DOI. And so, given the large dynamic range, FIREX-AQ really provides us an important opportunity, in addition to doing the science and characterizing the fires, but also to understand how the instrument performance varies under real-world conditions. With large signal to noise. Too often we're looking at very small signals, either in very clean remote marine atmospheres or in the upper atmosphere, and we have a lot of we're have difficulty making the measurements and teasing out the aerosol properties, and we don't always have great signal for actually evaluating the instruments. And so when we're doing a fire plume study where we're very close to the emission source, we obviously don't have that problem. And so FIREX-AQ is going to really prove to be really valuable for assessing the instrument performance under these really high aerosol loading concentrations. And so we'll look for a forthcoming paper that we're going to submit to Atmospheric Measurement Techniques, talking about the large suite of instruments and some of those properties. And so at this point, I'd encourage you to, this was the high-level summary, I'd encourage you to visit the FIREX-AQ website, given at that link below. And I thank you for your attention. And so for those who are interested, I'd like to show a little bit of data that's kind of in the weeds, talking about some of the aerosol measurements that we made and some of the consistency checks that we did for these different instruments. So if you're interested in that, please hang on for a few more minutes. And so the first set of measurements that I want to talk about are those optical properties, are scattering measurements. And so typically what we're doing here is we're using nephelometers to measure the scattering coefficient of the aerosol. And we want to understand how this scattering coefficient varies under both dry and humidified conditions, because we know that aerosol particles can uptake water and actually grow in size and scatter more light. But one of the challenges that we always have with this is that in order to dry the aerosols, we have to use either a silica gel or a Nafion dryer, and typically we use a large perma-pure multi-strand Nafion dryer to accomplish this. And there's always been some question in our minds about, well, what is the particle transmission efficiency through the dryer? And so prior to FIREX-AQ, we did some really detailed measurements in the laboratory to understand what the transmission efficiency is through this dryer as a function of size for different polystyrene latex spheres aerosols. And so what this graph on the right is showing is the results of that transmission efficiency for the membrane Nafion membrane dryer, given by the gray curve in terms of a number ratio, and the red triangles in terms of the scattering coefficient ratio. And what we can see is that generally we do fairly well up to about just a little less than a micron, and then we start to drop off as the larger particles are unable to penetrate through the dryer. And so this is very different from what we're expecting from our inlet. Based on past work, we know that the inlet is able to transmit particles in that are smaller than about 4 to 5 microns, given by those purple and green curves. And so this would result in a pretty unclear scattering coefficient depending on the contribution from the coarse particles. And so our solution is to use a cyclone. And so we also did the evaluation with the scattering transmission efficiency without the dryer in line using a 1 micron cyclone that's given by the blue curve. And you can see that that does a really good job of giving us a good constraint, upper constraint on the size of the particles contributing to our scattering coefficient really before that transmission efficiency starts to drop off. And so for FIREX-AQ, we incorporated a 1 micron cyclone upstream of our dry and humidified scattering measurements in order to give us more confidence in this measurement. In addition to our optical property measurements, we're also using condensation particle counters, CPCs, and optical sizers, like the laser aerosol spectrometer, LAS, in order to both count and size the particles that are coming in through our inlet. And so we wanted to understand, well, we have a number of different instruments that are making these measurements. We wanted to understand how well they agree and how that agreement varies with size, because we know that as particles get smaller and smaller, that can affect the ability of specific instruments to detect their concentrations. And so what these graphs are showing is they're showing the number concentration measured by the particle counters or the integrated size distribution from the LAS and our scanning mobility particle sizer as a function of the DMA classified dry mobility particle size. And we're ratioing that to the total concentration measured by one of the condensation particle counters, the ultrafine counter, a TSI 3776. And so what we can see here is that in addition to the drop-off that's very noticeable on the left graph at various cut sizes of the CPCs, we can see that there's some disagreement in the upper plateau. Once we get above those lower detection limits, there's some biases associated with the overall number concentration, and that by and large, most of the counters tend to be biased low compared to the 3776. However, we can also see that generally these counters agree to within about 10 to 20% of each other, although they're all undercounting relative to the 76, which might suggest that that one might be reading a little high. And so for FIREX-AQ, we chose not to make any changes to the particle concentrations that we archive, but instead noted that there's some uncertainty here on the order of about 20% between these counters. The only correction that we did make was with that red curve, the hot CPC. And so this was a pair of particle counters, one that was thermally denuded to 350 degrees C, that's the hot one, given by the red curve, and the one that wasn't, given by the blue curve. And we did scale the hot CPC up by about 6% to count for any thermal denuder losses that seem to be reflected by this calibration curve. Looking at the integrated number concentrations from the sizers, again, we can see very consistent with those particle counters and very consistent with each other. And so we chose not to make any corrections for these instruments. And I talked about that thermal denuder. We also did some very detailed measurements of the temperature profile in the denuder. And I've been saying that we set it to 350 degrees Celsius as our nominal set point. But of course, we know that the actual temperature that the aerosols are being exposed to in the tube probably varies somewhat differently from that. And so what we did was we used a thermocouple, a flexible thermocouple that we could shove through that tube and to measure the temperature profile along the length of the tube. And so our thermal denuder is about a 6-foot long stainless steel tube that we've wrapped into a coil to minimize the footprint that is on the aircraft. And then we've wrapped that coil in high temperature heating tape, which is temperature controlled at a nominal set point of 350 degrees Celsius. And so what the graph on the right is showing that temperature profile for a number of different particle sample flow rates through the tube. And so what we can see is that after an appropriate entrance length that depends a little bit on the flow, we get up to about a nominal temperature of around 370 degrees Celsius, so running a little hot. And that tends to oscillate plus or minus about 40 degrees Celsius. And the oscillations are damped as the flow rate through the system increases. And so this gives us good confidence that we don't have cold spots in our system, that we're able to heat the aerosols up to even actually higher than our nominal set point to ensure that we're removing and stripping off the volatile component of the particles. Finally, and I just want to note this is FIREX specific, we have a number of different particle sizers. That some of them are based on electrical mobility sizing, some of them are based on optical sizing, and some of them are based on mass spectral particle time of flight sizing. And so we wanted to assess, using FIREX again under these high signal conditions, the extent to which these different measurements agreed. And so I'm showing on the left here an example set of perpendicular transects through the smoke plume. This is for the Williams Flats fire on August 7th, 2019. And on the right, I'm showing the size distributions for different legs through that fire plume at points where the size distributions for the different instruments all agreed. And so in some cases, this is driven by the availability data for the AMS, which wasn't always running in size resolve mode. And so we're seeing a number of different size distributions. This is in number of particles, dn d log dp, plotted versus the particle diameter in nanometers. And we started at the upwind leg, very low concentrations. The size distribution is measured by the two optical sizers, the LAS operated by large, the SMPS that we operated, and the UHSAS, which was operated by the NOAA group, agree very well. And similarly, this is true for the downwind plumes as well, where we had good agreement between those three different optical sizers. In addition, we wanted to compare to the AMS sizing, and we see that agrees very well for the fresher smoke plumes, although we start to see some significant variability in the sizing associated with the LAS and the SMPS, and this may be due to the difference in sizing time that it takes the SMPS to do a 60-second scan versus a 1-second scan for the LAS. And so by looking plus or minus a few seconds on either side of the comparison period, we can see that there's a significant amount of variability in the size distribution, which may explain that disagreement. But this gives us really good confidence that these instruments are all measuring the same thing and giving us the same response. So I'll just end with the same summary slide that I showed before. We've done a lot of work to evaluate the consistency of the different particle sizers and counters and optical measurements. And we're looking forward to doing more science. So thank you for your attention.